My name is Maddie Szoo. I'm a fourth-year chemical engineering and biochemistry student at Northeastern, and today I'm going to talk a little bit about my experience as a co-op in the CaNCURE Nanomedicine program. So my experience took place at the lab of Professor Tayeba Hassan, which is at the Wellman Center for Photomedicine at Massachusetts General Hospital, or MGH. And this is a photo of all of the Wellman Center at our annual retreat, including my lab mates and Professor Hassan at the front here. This experience is really positive overall. I was able to network and gain a lot of professional connections. And because the lab was on MGH's main campus in Boston, I was exposed to a lot of different scientific talks and career development opportunities that the full-time employees were able to take advantage of. So that was really positive as well. Day to day, it was very self-paced. I gained a lot of practice in experimental design and planning with the help of my mentor, as well as other postdocs in the lab. And this really reaffirmed my interest in cancer research overall. My project specifically focused on the photodynamic priming of pancreatic tumor spheroids for enhanced immune checkpoint therapy. And I'm going to briefly talk a little bit about my project background and some of the things I was able to do. So as some background, pancreatic cancer is currently the third leading cause of cancer-related death in the United States today, and the subtype of pancreatic ductal adenocarcinoma, or PDAC specifically, has a lot of challenges due to the presence of desmoplasia, which is the formation of fibrous connective tissue in response to certain malignancies, including cancer. And this desmoplastic response is really challenging to treat because the presence of those fibroblasts and other extracellular matrix components creates a dense stroma, which reduces drug distribution and efficacy. Our lab specifically focuses on photodynamic therapy, or PDT. As an overview, photodynamic therapy is a potential cancer treatment option in which a non-toxic photosensitizer is exposed to cells, and after a certain incubation time, and that incubation time may vary based off of the formulation and type of photosensitizer, the photosensitizer is exposed to light of a certain wavelength through the use of a laser, and that helps to excite the molecule and cause the formation of cytotoxic reactive molecular species, including singlet oxygen, which then kills the cancers. So PDT has a lot of versatility, and it is helpful in resensitizing therapy-resistant cancers, including those refractory to immunotherapy, which pancreatic cancers typically are. So photodynamic priming is a subtherapeutic form of PDT, and this is really useful in increasing the permeability of the tumor microenvironment and breaking up that dense stroma that I talked about to help improve drug delivery. And it's also useful in producing immune stimulatory signals, such as damage-associated molecular patterns, or DAMPs. These DAMPs are really important in the cancer immunity cycle. They are released by dying tumor cells in response to treatment, and the DAMP that we focused on specifically in our study was calreticulin. The antigen-presenting cells, which are immune cells, then engulf the dying tumor cells that are expressing calreticulin and present the cancer antigen to activate and train the T cells to recognize these cancers off-site. However, the tumor cells evolve and express the PD-1 ligand to inhibit this T cell activity, so it is immunosuppressive in nature. The anti-PD-1 antibody is an immune checkpoint inhibitor and a form of cancer immunotherapy that helps to overcome this issue. It binds the PD-1 surface receptor on immune cells, blocking the PD-1 ligand, which is the immune suppressive signal secreted by the tumor cells. And this helps for the immunogenic cell death pathway to proceed. The objectives of my project specifically were first to optimize PDT dosimetry to enhance immunogenic cell death in our in vitro PDAC spheroid model that we created. And second, to establish enhanced responsiveness of PDP-treated spheroids to this immunotherapy with the use of a peripheral blood mononuclear cell co-culture, or an immune cell co-culture. And this is a diagram kind of outlining the steps of our project and the creation of the spheroids and the co-culture with allogeneic PBMC samples. This is actually a real image of our model from the lab, and we have M. cherry expressing MIA-PACA2 cancer cells, as well as the GFP, or green fibroblasts, and this is to help mimic the different desmoplastic conditions that you'd see in patients. Typically, you can see desmoplasia make up between 10 and 90% of total tumor volume in pancreatic cancer patients, so we wanted to vary the ratios of 3:1, 1:1, and 1:3 to see how these different desmoplastic conditions would respond to our treatment. This impact on cancer nanomedicine has the potential to be very relevant. It demonstrates the novel applications for photodynamic therapy and the utility of photodynamic priming and combination therapies for PDAC. And our anticipated outcomes of this project are to develop this clinically relevant desmoplastic PDAC model with varying stromal compositions, to determine the lethal light dose for PDT, as well as the subtherapeutic dose for PDP that's specific to our model, and also to optimize the co-culture of these PDAC spheroids with the allogeneic PBMCs that I was talking about to really understand the immune cell response. This is useful for future immunotherapy studies, as well as to better understand anti-PD-1 dosage in the clinic. So the skills and knowledge that I gained were really based on photochemistry, which is the focus of our lab. I learned about the theory and practice of PDT as a possible treatment option for pancreatic cancers, but also for anything from antibiotic resistance to oral cancers, where there were other projects that were also going on in these areas in the lab. I also developed a deeper understanding of immunotherapy and performed assays exploring combination therapies targeting different stages of that cancer immunity cycle that I talked about before. I gained experience in the culturing of the 3D immune organoids, which was not a model I talked about, but another model I was able to assist with, as well as with the spheroids that I showed you in the last slide. Lastly, I studied pancreatic cancer biology, and I learned the challenges to potential treatment options for patients today. My future goals, what I would like to learn about more is how biomaterials can be used to mimic the tumor microenvironment. I was really interested by the different 3D tumor models that we were creating, so I'd love to learn more about how to optimize those in the future. And for my next co-op, I'm hoping to pursue either another academic or potentially an industry position that focuses on immuno-oncology or tumor modeling similar to this. My most exciting event in my co-op is attending the ASBC EMBO conference, where I was able to present this work on the spheroids. And this experience really reaffirmed my passion for cancer research, and it gave me insight into the applications of tumor models in personalized medicine, and helped me kind of see what other labs were working on in a similar area of research. So thank you to the Hassan Lab and to all the CaNCURE staff for this experience. I really appreciate everything that you put together, from the talks to the meetings. These are my citations, and have a good day.